Today I will be talking about cell cycle, and first off, I would like to talk about this piece right here, which is the yellow piece, which is considered interphase. First, I will be talking about G1, also known as gap 1, which is the cell growth and interval between mitosis and DNA replication while the cell grows metabolically but does not replicate DNA. And then secondly, I would like to talk about the S phase, which means synthetic, and that is also DNA replication. Uh, it's a DNA replication occurs doubling its amount, and then chromosomes, that number remains unchanged from 2n to 2n. And then it is in an animal cell, which the DNA replication in nucleus, centriole duplication is in the cytoplasm. Lastly is G2, which is also known as GAP2, and it is the preparation for mitosis and is also protein synthesis and for cell growth to continue through the preparation of mitosis. Next thing I will be talking about are these four things right here, or five things actually. This purple part is known as mitosis. P stands for prophase, M stands for metaphase, the A stands for anaphase, and the T stands for telophase. So prophase is actually the first stage of mitosis, and the chromosomes condense into visible structures and the nuclear envelope breaks down as the mitotic spindle begins to form. And then second would be the metaphase, which would be the second stage in the cell division, and is where chromosomes align along the center of the cell at an imaginary plane called the metaphase plate. Then the third phase would be the anaphase, which is the third stage in cell division, and where the sister chromatids are pulled apart and moved to opposite poles of the cell. And then telophase would be the final stage of cell division and mitosis, and where two nuclei form around the separated sets of chromosomes at opposite ends of the cell. Next will be the cytokinesis, which is not part of mitosis. It is on its own, and it is the final stage where the cytoplasm and organelles of a single parent cell are split into two separate daughter cells. And then we move on to the phases of mitosis, which is called interphase, and that would be this image right here. That's how the image would look. And then secondly would be prophase. As I said, in interphase, it is where the cell grows and replicates its DNA to prepare for cell division, while the stage occurs before mitosis commences. Then as we move on to prophase, the chromosomes condense and then the nuclei disappears. So then we move on to metaphase, which is where the chromosomes align in the centriole of the cell and attach to microtubules in preparation to split apart. While moving on to anaphase, they attach together and the chromatids separate and move to opposite ends of the cell. This stage ensures that each cell contains identical chromosomes after splitting. Then we move on to telophase, which is the last stage of the interphase and mitosis. And the telophase is where two nuclei form around each set of chromosomes, and chromosomes decondense and the cell begins to split from each other. Then at the very end, we have cytokinesis, which is where the cytoplasm of the parent cell splits to form two identical daughter cells.